what a pleasure to have a great lineup of speakers today. Couple of reminders that I will just mention would be that outside of the speakers, if you could please go ahead and leave the cameras off and go ahead and mute the speakers as well, or the microphone as well. And that will just help us in terms of keeping the talks moving forward. And then for questions and answers, after each of the presentations, we will have a few minutes to do Q&A. So please go ahead and post your questions in the Q&A box or in the chat box. I'll be tracking those and I'll help to moderate those. So without further delay, it's a pleasure to first introduce Dr. Elaine Stur. She is a postdoctoral fellow. She obtained her PhD degree in Brazil and has been doing really elegant work in terms of looking at single cell as well as spatial analyses in ovarian cancer. And her goal has been to understand and overcome immune suppressive pathways in these cancers. With that, I'll turn it over to Elaine to start her presentation.
Good morning, everyone. It's such a pleasure to be able to be here today and present a little bit of our work. Thank you, Dr. Sood, for the introduction. So today, I'm going to present a bit of the work that we have been doing with ovarian cancer that we call Resolve the Spatial Transcriptomics of High-Grade Serous Ovarian Carcinoma. So to start with, I'm going to give a short introduction about ovarian cancer, followed by some of the results that we obtained, and finalize with a Q&A. So epithelial ovarian cancer is a very heterogeneous disease with many different subtypes. So the disease is considered heterogeneous to start with because present many subtypes, being the most common one, the high-grade serous ovarian carcinoma. The disease presenting about 1 in every 75 women that will be diagnosed with ovarian cancer during her lifetime, and 63 is the average age of diagnosis. So according to the disease, the risk factors for ovarian cancer are also very heterogeneous. So in general, all women, mainly with older age, are in risk for ovarian cancer. So family history is also important, ethnicity, genetic mutations, being about 98% of all ovarian cancers, they present P53 mutations. Also, very often the patients present BRCA1 and BRCA2 mutations. But a very important point that high-grade serous ovarian carcinoma actually is a disease that is highly characterized by the presence of CNVs, of copy number variations. The reproductive history is also important as well, hormone replacement and other factors, for example, endometriosis. So one issue with ovarian cancer is that the detection is not normally made in the early stages. So basically there are not really strong detection methods for ovarian cancer. For example, a Pap test is not able to detect ovarian cancer. So due to this fact, it's very common for the disease to be detected in already stage 3 and stage 4, with only about 35% of the women diagnosed with late stage disease will survive 10 years or more. So the treatment of disease depends a little bit of the histology of the disease, the subtype, but mainly is done by surgery, by chemotherapy that include carbotaxol, sometimes target therapy like Avastin, PARP inhibitors, and I would say the hot of the moment now, it is immunotherapy, but unfortunately, we do not have a good response of these patients to immunotherapy. Only about 20% of ovarian cancer patients, they really will respond to the immunotherapy. And that's quite interesting, actually, because there are so many studies trying to understand not only how immunotherapy works for cancer, but for ovarian cancer specifically. And it's not really clear why these patients, they don't respond well. It's known, for example, that ovarian cancer, in fact, has the infiltration of CD8 cells and many other subtypes of immune cells. But even like that, we do not observe good response. So again, as I have been saying since the beginning, the heterogeneity of disease, we also can observe that on the response to therapy. This graph, we can observe here 4 different groups of patients. Those patients that goes first to the neoadjuvant chemotherapy and achieve or not complete resection at the surgery. So R0 means achieve complete resection, R1 does not achieve, or those patients that goes directly to primary surgery, achieving or not the complete resection. And we can observe actually that these patients, they have a very different progression-free survival. For example, patients that goes to primary surgery and achieve complete resection, they're gonna have around 48 months when you compare with patients, for example, that goes first to NACT and does not achieve complete resection. And again, we don't know exactly why this happened, what leads to such a different response on this type of these patients. That's one of the reasons why it's so important for us to understand more about the biology, about the molecular biology of this disease. So even with our group, we have for quite some time now trying to understand what are the difference between those patients that needs to go first to neoadjuvant chemotherapy or those that does not respond and those that respond well to this therapy. What is the difference between them? So in 2020, our group published a paper that included many different groups and many different omics, including transcriptomics, DNA analysis, proteomics, phosphoproteomics, immunoprofiling. And actually, yes, we did observe some difference, mainly in CNVs and T-cell infiltration. But actually, this difference that we observed was not extensive as we had anticipated, considering how different these groups are. So, and these finds, they suggest actually that there are important molecular differences between such tumors, but unfortunately we are not able to understand only analyzing the bulk molecular of these tumors. So that's the reason why we use the in-depth spatial analysis to explore the heterogeneity of high-grade serous ovarian carcinoma and also to determine how the dynamic behavior between the tumor and the tumor microenvironment varies between poor and excellent responders to neoadjuvant chemotherapy. And to generate this data, we performed the deep spatial transcriptomics profiling in situ. So that's just a brief summary of the analysis that we perform. Basically, we decided to use these 2 groups, the poor responders and the excellent responders to neoadjuvant chemotherapy, and perform spatial analysis. So here, it's our study design. So through the Moonshot program, the patients, they go to laparoscopic assessment, and then they are classified in R0 feasible, so those patients goes to tumor reduction surgery, or R0 not feasible. So, and then those patients, they receive neoadjuvant chemotherapy, and later they can be classified in poor responders. There are those patients that has stable or progressive disease after 3 or 4 cycles, or suboptimal interval cytoreduction after NACT, or the excellent responders. There are those patients that achieve complete response or only microscopic disease left at the time of the interval surgery. So we are focused on those patients that goes to neoadjuvant chemotherapy, and later we could classify them in poor responders or excellent responders. That was a pilot study, so we had 6 patients in each group. So the sample collection was tumor biopsy from the primary site and from chemo-naive patients, and we performed the spatial transcriptomics, and we did clustering analysis, cell type identification, pathway analysis, bulk sequencing comparison, and geographic location measurement, and ligand receptor analysis. So to perform this analysis, we use the 10x vision spatial gene expression. So the interesting of this methodology is that allow for us to explore the tissue architecture. So we don't need to do a tissue dissociation or anything like that. We can use a slice of our tumor and perform the spatial transcriptomics on that. So here, one advance in comparison of the single cell, for example, would be that we don't need to do the cell dissociation. So we can maintain the tissue as it is. One point that I would say maybe is not so strong, it's because unfortunately it's not at single cell level yet. But here, for example, we have one real image of one of our samples. These were tumor biopsies, so that's why they are quite small. And this is one of the capture areas of the Visium slide. So they have a 6.5 to 6.5 millimeter area. So we need to do the mounting of the tissue in our slide. So, and here is one of the biopsies. And then we do the H&E staining on those samples before performing the spatial transcriptomics. So first we need to do the tissue optimization to guarantee that we're gonna have a good barcode binding with the RNA present on those tissues. Then we do the H&E staining, the imaging, the library construction, and the data processing. So now going a little bit deeper on our results. So the first image here show a comparison of the H&E imaging and the cluster distribution. So to start with, we kept all the 12 patients together. So, sorry, I have a little bit of sore throat issues here. So, and then you do the H&E imaging. As you can see here, this is our sample. And then we put all the 12 samples together to start with to identify the clusters. So basically the clusters, they were created based by molecular similarity. So we didn't, at this point, we did not define cell type or any specific molecular characteristic of this tumor. It was an unsupervised clustering. And what is interesting here, we can observe, for example, these brownish areas and these pinkish areas. They really match more or less on the stromal areas. So meaning that even using this unsupervised clustering, we are able to identify areas of the tumor, for example, stroma or epithelial areas. So next, after doing this rough cluster identification, we are able to identify 9 different clusters when we have all the 12 samples together. The next step now was to separate them on the ER, the excellent responders, or in the PR, the poor responders, to start to identify the difference between them. So here we have a graph showing the proportion of the clusters by ER and PR group. And we can observe actually that clusters 1 to 4, they were largely represented in both groups. But we also observe actually that certain groups, they were identified in one group, but not necessarily in the other. Like for example, cluster 7, that was identified on the PR group, but was not identified on the ER group. So here we show the quantitative representation of cluster proportion by each sample. And again, we can identify that not necessarily all the samples will contain all the clusters that we are able to identify. Only clusters 0 to 4, again, were largely represented between all the samples and between both groups. So next step, we try to do the cell type identification here. So knowing that in single cell analysis, that's a common type of analysis should be done to be able to understand a little bit more the tumor microenvironment. So here we perform the same type of analysis, and we identify at least 20 different cell types and 2 major areas that we call immunity CD45, representing more or less all the immune cells, or the stroma areas, representing largely the multiple cell types of the stroma. And one thing that is important to highlight here, we don't see clear cell types by cluster, those are the clusters, because each cluster, it is in fact represented by more than one cell type, as we did the unsupervised clustering and not the clustering by cell type specifically. So the major things that we could identify here actually is the PR group, the poor responders. They are stromal-dominated groups. We can see a lot of CAFs and myofibroblasts, and even this large stromal area being highly represented on the poor responders group. In the opposite, in the excellent responders group, we see major immune-related areas like B cells, T cells, and even this immunity CD45 being more represented in the ER group than in the PR group. So we also observed the epithelial mesenchymal transition-like cells in the most clusters of the PR group when we compare with the R group, showing that PR group, they are more largely represented by stromal-dominated cell groups in the opposite of ER group. There are more immune-related areas. So the next analysis that we did was try to understand how much the spatial transcriptomics would add to the analysis that we had previously performed with our book RNA sequence. All these samples that has the spatial transcriptomics data, they also have the book sequencing done that is represented here on this last line. So we call cluster all the combination of all these the clusters 0 to 4 from the spatial transcriptomics data. We chose these clusters because they were largely represented in all the samples. And here on the first line is the book sequence. Exactly the same samples, but now in a real book sequence. And actually, we observed that in the book, we really have this blending phenomena where this mixing, we're getting a book of tumor and doing the transcriptomics on that hides deeper profiling of the tumor, in addition to destroy the geographic localization of the tissue. So we cannot understand small specificities of those tumors. And some of these examples we can observe looking at in a few genes. So these were genes that were collected from Cosmic. So there are genes that are well known to be changed in ovarian cancer. In some cases, for example, like NF1 or MSH6, we see that when we look at the book sequence, we see upregulation of these genes. And when we look at this cluster all that is basically mimicking the RNA book sequence, we see the same trend. But it's interesting because when we look now into specific clusters, they don't necessarily follow the same trend. Some clusters we see downregulation of one gene, and other clusters we see upregulation of the same gene, meaning that different areas of the tumor, they will in fact have different patterns of one same gene. Other gene, for example, is BARD1. BARD1, it's shown to be downregulated on the book, and in our cluster altogether, we see an upregulation of the same gene. And again, when we look into specific clusters, when we look into specific clusters, for example, cluster 0, we also see an upregulation of these clusters. All the others show a downregulation. And what was interesting here, actually, that when we look at the real physical size of the cluster 0, based on the unsupervised clustering, the physical size of cluster 0 is much larger than the other clusters, meaning that possible the reason why when we put all these clusters together, we do see a final upregulation here, it's because the highly representation of the cluster 0 on these samples that we are analyzing here. So, and for example, we are not able to see this when we look at book sequence. In this case, we just knew this gene was downregulated, but we didn't know if it was in all the tumor or specific areas. So, and was very clear also on the pathway analysis that we performed the same type of comparison. Here we have PR and ER book, exactly like was performed before, and we observe exactly the same trend. So for some pathways, we are seeing an upregulation in the book and a downregulation of all the clusters together, or even the opposite, showing opposite trends, one showing upregulation, the other showing downregulation, and we observe exactly the same thing again on when we look at clusters. They have different clusters. So then we are able to identify this variation of patterns between book and spatial transcriptomics, meaning the technique, the way that you analyze, the characteristics of the tumor that you can keep, it's really going to be important for you to be able to identify, to do their full characterization. But also, and I would say even more important, we are able to see that when we are looking at different clusters, we do observe that different areas of the tumor, they have a very different pattern of gene expression. So, and that's quite strong. That is enough to modify even the pathways related with them. So as one thing that we have been focused, it is on the immune suppression of these tumors. We decided to look a little bit deeper on the immune-related pathways. So we did a selection of some pathways to see better. And here we are seeing the pathways being up or down on the PR group. And according to the result that we observed in the beginning, that the ER group actually, they have more immune-related areas, was interesting to see that, in fact, we see on the PR group a downregulation of multiple immune response pathways. So, in fact, proving again the data that we observed in the beginning, that the immune system, we see this more immune activation on the excellent responders, which does make sense with the previous data showing the literature. But then what was also interesting, actually, that when we look at this cluster 4 here, we see the opposite from what we are seeing from the other clusters, because actually we are seeing an upregulation of almost all the immune pathways related here on the PR. And so we tried to understand the reason why this could be happening. Actually, when we went back and looked the immune cell type characterization of these clusters, we can identify a lot of macrophage, dendritic cells, neutrophils, so which does make sense for us to see a upregulation of these innate immune pathways here. So the next analysis that we performed was the physical distance between cluster. So as you may remember from the beginning, so the clustering was done in an unsupervised way, meaning one same cluster, they can be found in many different areas of the tumor. So they are not necessarily all physically together, because we decide to cluster based in molecular similarity, so not necessarily in the physical connection between them. And then on this step, we try to understand what could be the connection of these clusters. So, and then we identify actually that this physical distance between clusters, they are largely different between groups, as we can observe on this line here that shows basically the same comparison. So we can observe actually that for example, the same clusters, they are more physical closer on the ER, meaning, for example, the cluster 0 has a tendency to be physically closer on the ER group and a bit more far away on the PR group. And we can observe this same trend for multiple different clusters. But then we also observed that clusters that were molecular similar, they were, for example, on the ER group, the cluster 5 and 2, they were physically closer to each other than the same cluster 5 and 2 on the PR that were more physically far away from each other. And we also observed the opposite, like clusters like 8 and 3, they were closer on the PR, and on the ER, they were more physically distant. And why this is interesting? Because on the cluster 8, for example, we see a large representation of the fibroblasts. So, and again, we saw more stromal areas on the PR group. So basically, we can see that this different cell-to-cell contact, more than the disposition of a cell type, it could, for example, help to explain different mechanisms of response to different therapies. The last analysis we performed was the ligand receptor analysis, where we compare the ER group and the PR group. So we use all the pairs initially described in the literature, and right away we can observe that we have multiple pairs that are pretty different when you compare ER and PR. And what's interesting to see here, that we got some pairs, for example, GPC3, EGF1R, and when they are physically closer, their score is higher, meaning they have a stronger connection than when you compare with 8 and 2, for example, that is far away on the ER group. And doing the same type of comparison from the PR group, we observe exactly the same trend, meaning that clusters, ligand receptors from clusters that are physically to each other, they have a higher score, which could be related with better relation and connection between these pairs. So the most important findings here is that the excellent and poor responders, they show different composition of the tumor microenvironment, with the poor responders being more stromal-derived cell populations. So we do see a variable regulation of intercluster cancer-related genes and functional pathways inside the same group of tumors. So one same tumor can show these very different regulation depending on specific areas. The geographic location of cell populations, they help to define the cell-to-cell communication. And this is one of the first application of spatial transcriptomics in high-grade serous ovarian carcinoma. So I just would like quickly to thank you all the collaborators that we have for this project, including my mentor, Dr. Anil Sood, and all the other labs that help us, like Baylor and other labs and MD Anderson as well. Thank you so much.
Thanks so much, Elaine. There are a couple of questions for you. One is what is R0 and its significance? And there are a couple of other questions as well.
Okay, so the R0 means those patients, they go to surgery and they are able to achieve complete resection, meaning they have no visible disease. So, and it's mainly important because we do see that these patients in a long term, they will have, for example, different progression-free survival. So that's why it's important to define those groups.
Second question, Elaine, is how do you determine downregulation of pathways using GSEA? Do you mean that the pathways were not enriched?
So they were enriched, but then when you perform this type of analysis, so I don't know completely the pipeline, the math behind it, but you have a combination of genes that when you use these well-known pathways, you can infer if this pathway should be upregulated or downregulated based on a combination of genes and how those genes are being altered inside a pathway.
Okay. And I think this question you may have in part addressed already, but can you elaborate on characterization of clusters? What cells do they predominantly represent? And did you see changes in cluster abundance between ER and PR?
So yes, for the cell population, we do observe a strong difference between them. So to start with, when we did the unsupervised clustering, we did not know which cell type we could identify there because we focus more at first on the different patterns of gene expression. So that's first how we define it. And then second, we decide to look at the specific cell types. And then we observe that all the clusters, basically, they have all the presence of the multiple different cell types. We observe a lot of epithelial cells in basically all the cluster, a lot of presence of different immune cell types. But then when you want to see the difference between ER and PR, actually, we observe that the PR, they have more stromal areas, meaning we are observing a stronger pattern of stromal cells, mainly calves, on these PR patients.
And then the final question, Elaine, is it was interesting to see more DNA repair genes upregulated in PRs. Why do you think that is?
So we have discussed a little bit about this. So I'm not really sure why we are observing that, but could be, for example, I don't know the best way to answer that. Okay. I'm sorry.
Okay, I think that's the questions, Elaine. Great job. And let's move on to our next speaker, who is Dr. Melanie Winkle. She carried out her doctoral work in the Department of Pathology in Netherlands on the role of non-coding RNAs in B-cell lymphomas. And then she has been carrying out her postdoctoral work with Dr. Goerge Calin here at MD Anderson. And today she will be speaking on antimiR 93.5p therapy prolongs sepsis survival by restoring peripheral immune response. Melanie, I will turn this over to you. Welcome.
Thank you so much for that kind introduction. I'm happy to be here. Thank you for giving me the opportunity to share our work today. There we go. So I will jump right in. I will start with a short background on sepsis and then present the results of our study. So sepsis is defined as an organ dysfunction caused by a dysregulated host response to infection. What this means is, upon recognition of the infection, a progressive immune response is triggered that is characterized by both hyperinflammation and immunoparalysis, leading to progressive tissue damage. So here we have activation of both pro and anti-inflammatory pathways. And in the end, this can lead to multi-organ dysfunction. So the elderly and immunosuppressed are especially prone to sepsis-associated mortality, as this immunoparalysis component is very strong in these patients, leading to deterioration rather than recovery and clearance of the infection. And currently, there are no efficient therapies available for the treatment of sepsis. So sepsis in cancer patients accounts for a large part of sepsis cases. About 15 to 20% of ICU admitted sepsis cases are cancer patients, and the cancer-related sepsis has a higher mortality rate compared to a non-cancer related sepsis. And in cancer, of course, we have both cancer and treatment-related immunosuppression already present. So from the treatment, we get defective phagocytic activity, neutropenia, lymphopenia, general immunosuppression. But the cancer itself also has an immunosuppressive microenvironment. So if we look at the healthy state, where we usually get interaction between APCs and T cells via a CD28 and T cell receptor stimulation, which then results in proliferation and the downstream activation of other immune cells. In sepsis, this is disturbed through immunosuppressive mechanisms such as CTLA and PD1L ligand interactions, leading to apoptosis, energy, an upregulated regulatory response, and Th2 polarization, and then defective phagocytosis. In cancer, this immunosynapse situation looks very similar to what we observe in sepsis, and this also leads to a T regulatory response and defective cytotoxicity for cancer cells. So cancer patients are kind of primed for immunosuppressive phenotype through both therapy and cancer-related mechanisms. So in our study, we hypothesized that microRNAs deregulated in the immune cells of sepsis patients are involved in the disease progression. And we aim to identify and functionally characterize microRNAs that qualify as therapeutic targets for the treatment of septic patients. So to do this, we took 4 major steps. Starting with target selection, we studied the peripheral blood mononuclear cells of sepsis patients and controls to see up and down regulated microRNAs, and also did a murine sepsis model called cecum ligation puncture-induced sepsis. We then confirmed our targets by looking at the specific expression of these microRNAs in immune cells, and also applied a sepsis model in baboons, which was E. coli or S. aureus induced. We then performed therapeutic studies using an anti-microRNA and did a survival analysis in this murine CLP model. And in the end, we did functional studies using microRNA modulation. So we looked at the microRNA, the effect of antimiR therapy on blood cell composition in these mice. And we also identified microRNA target genes in miR93 knockout cells. So to the first part, which is the target selection, here you see a heat map of microRNA expression in sepsis and control cases. Control cases here are post-surgery patients, and healthy is defined as the absence of infection or known medical conditions. And here we find 4 microRNAs that are significantly upregulated in these sepsis patients, namely 182, 486, 16, and miR93. We then moved on to a mouse model, and this work was performed by my collaborators Michna and Enrique at the MD Anderson Cancer Center. We did the cecum ligation puncture-induced sepsis in mice. And when we induce sepsis in this way, we see upregulation of multiple pro-inflammatory as well as anti-inflammatory cytokines, as is a hallmark of sepsis. And when we look at microRNA expression in the sham compared to the CLP-induced sepsis, we see all 4 microRNAs are significantly upregulated in this model. For target confirmation, we then looked further into immune cell expression. Firstly, we studied expression of the sepsis microRNAs in 5 immune cell types from 10 healthy donors. This is based on a publication. And while miR-182 is very lowly expressed in all these cell types, we see significant expression of miR-93 and very high expression of miR-16 as well. We then had a closer look at T cells, and this was done in collaboration with Katie Resvani's group. And here we see, so we couldn't measure miR-182 in T cells, and the other, so what you see here is resting and activated T cells, as we see interferon gamma increase when they're activated, and miR-486 as well as miR-16 have stable levels, and miR-93 shows a strong increase in the activated T cells, showing that this microRNA is likely to have an important function in T cells. We then went further to study miR-93 expression in a baboon sepsis model. This was done with collaborators from the Oklahoma Medical Research Foundation. And here we see in sepsis induced that was induced either with E. coli or S. aureus injection in these animals, we see a steady increase of miR-93 in these animals upon sepsis induction. And I've noticed that the animals that experienced an early death actually showed much higher upregulation of miR-93 compared to those animals that survived a longer period of time. So there's a correlation with the severity of the sepsis. We then went on to therapeutic studies using antimiR treatment. So here we have 2 groups. Both are have CLP-induced sepsis and are treated either with a control, with a scramble, or with anti-microRNA-93 therapy. And upon giving this treatment, we see strong reduction in miR-93 in both plasma and tissue of these animals. And the cytokine levels that we saw increased upon induction of sepsis are decreased by administering this therapy. So we did this study in mice of different ages. We used young mice, 4.5 months old, intermediate age of 8 months, and somewhat older mice of 16 months. And what we see is that the survival is improved by anti-miR-93 therapy, and this effect gets stronger as the mice get older. And here is all the mice combined. So we see a significant effect on the survival of these mice. So next we looked at how the blood cell compositions change in these mice in response to therapy. So in this experiment, we have 4 different groups. We have control, sham, operated. So they're operated but do not have sepsis, and we have 2 sepsis groups treated with scrambled or anti-miR therapy. So the strongest effects we saw on immune cell compositions is, on the one hand, a very therapy-specific increase in CD4 and CD8 T effector memory cells. So these are T cells that actively fight the infection. And on the other hand, we found decreases in PD-L1 expression monocytes and macrophages, which was specifically induced in the sepsis group and decreased by therapy. And so this is summarized here. Looking at all the blood cells, we see so the therapy inducing a pro-inflammatory response in T effector memory cells, both CD4 and CD8, as well as this decrease in PD-1 ligand monocytes and macrophages, which would be immunosuppressive. So this also has a pro-inflammatory effect. To further study how microRNA 93 mediates these effects, we created a knockout cell line using CRISPR. So we used 2 single guide RNAs to cut out miR93 specifically, which is located in the intron of MCM7 in a cluster together with miR25 and 106B. So using this CRISPR, we get a specific knockout of miR93, while the other 2 microRNAs are not significantly affected. Neither is the level of MCM7 itself. Cell morphology did not change significantly, and the growth rate of these knockout cells was slightly lower compared to the parental and CRISPR control cells. These cells we then use to perform what is called AGO2 RNA immunoprecipitation. So this method uses antibodies against AGO2. AGO2 is a major component of the RISC complex in which microRNAs interact with their target mRNAs. So by pulling out this RISC complex together with the attached mRNAs, we can define which mRNAs are targeted by microRNAs in the cell. And by then comparing the miR93 positive cells to the miR93 negative cells, we can define the miR93 specific targetome. So doing this AGO2 RIP-chip, we defined the targetome of miR93. And firstly, what you see here is we cross-check the genes that we identified as miR93 targets using miR-tarbase. And the first hit we get is miR93, so there's really a good identification of the targets this way. And actually the first 5 hits that we get here are all part of the miR17 family, which share the same seed sequence. So this confirms that the AGO2 RIP really identifies miR93 targets. To further identify sepsis-relevant targets, we then overlapped these genes were identified in the AGO2 IP with the genes that were downregulated in our Baboon model. If you remember from the beginning, the miR93 was upregulated in the Baboon model, so its targets should be downregulated. So we have the E. coli and the S. aureus Baboon model, and here you see the overlap with these AGO2 RIP-chip defined genes, as well as predicted or validated targets of miR93. So if we do a pathway analysis on these genes that are affected in baboons, we see a very strong enrichment of multiple genes that are very important for T-cell receptor signaling, such as ZAP70 and CD28. Oh. There's something wrong there in the animations. So what you see here is the target gene expression in these miR93 knockout cells. You see the expression in the 2 knockouts compared to the control, which is SET21. And we see, for example, this high upregulation of CD21 when miR93 is not present. CD28, I'm sorry. And target gene expression in the baboon sepsis model, here you similarly see that upon induction of sepsis in these animals, CD28 goes strongly down. Also, for example, ZAP70, which is a very central molecule mediating T-cell receptor signaling. So if we go back to this situation where the interaction between APCs and T cells, miR93 inhibits this interaction of miR93 as well as TCR through ZAP70, causing immunosuppression and the absence of this activation of these downstream immune effects. So in summary, in sepsis, we have high levels of miR93, which will bind to its target genes. Here, I listed just a few of these, all involved in T-cell receptor signaling. This causes downregulation of these T effector memory cells fighting the infection and causes immunosuppression and worse survival. While if we administer anti-miR93 therapy, these genes can be upregulated, and this will restore the peripheral immune response and help to clear the infection. So what I've shown you today is that miR93 is upregulated in the PBMCs of sepsis patients, as well as in mouse and non-human primate models of sepsis. Anti-miR93 therapy prolonged the sepsis survival of sepsis mice, especially mice of an older age, which are more prone to immunosuppression. And anti-miR93 therapy restored the peripheral immune response by having pro-inflammatory effects on both T cells and macrophages. And miR93 targets very essential genes in T cell receptor signaling, such as CD28, causing immunosuppression. So as to future perspective, we are at the moment further studying the miR93 targets and its functional effects of miR93 inhibition in macrophages. And in addition to this, we're screening for small molecule inhibitors of miR93 to give a more better treatment options for sepsis patients. And with that, I would like to thank all the people that were involved in this research from different departments of the MD Anderson Cancer Center, as well as different universities all over the world. Thank you. And thank you for your attention. I'm happy to take any questions.
All right, great job, Melanie. So first is the first and third questions are actually same type of a question. They say, awesome data. What was the chemical composition modification pattern of the antimiR, and how was it delivered therapeutically?
So we used liposomes for the delivery of this antimiR therapeutic. I'd have to look up what the exact chemical modifications are on this specific antimiR. But so we used liposomal delivery in the mice that were injected intravenously.
Okay. Mm-hmm. And there's another question. Do you have data on the level of antimiR in T cells in the mouse model?
The level of, sorry, can you repeat that?
Do you have data on the level of the antimiR in T cells in the mouse model?
No, we do not have data on that. So we checked the expression in tissues as well as in plasma. We did not look in the PBMCs specifically. Yeah.
Okay. Mm-hmm. And let's see, here's another question. What does anti-miR93 do to immunotherapy resistance?
That's a very good question, and that's also what we like to what we're trying to follow up further, because we saw this PD-1L decrease in the macrophages, so that's why we're currently also looking at miR93 targets in the macrophages. So our data indicates that the upregulation of miR93 actually causes a decrease in the okay, let me order my thoughts. The therapy induced a decrease in PD-1 ligand expression in macrophages and monocytes. So it is possible that this checkpoint is indeed affected by the therapy. We don't yet know how, so we're trying to figure that out at the moment. But these PD-1 checkpoint inhibitors are also actively being tested in clinical trials for sepsis because there's this strong involvement of this checkpoint in the pathology. So targeting this additionally with anti-miRs would be a great opportunity therapeutically. Mm-hmm. Sorry.
And there's one more question here. How did you confirm the specific off-target or specific or off effects of targeting microRNA?
I'm sorry, I don't quite.
I believe the question was probably trying to get at, like, how perhaps differentiating, on versus off-target effects of the microRNA.
Mm-hmm. So by using this AGO2 RIP chip method, and by using this in knockout cells and normal cells, we specifically defined which mRNAs are actually interacting with miR-93 in the RISC complex. So those would absolutely be expected to be direct targets and targets that are inhibited to a significant extent in these cells.
Mm-hmm. Yeah. And this looks, I think, more like a comment. It's probably related, I think, to the question related to the immunotherapy, mm-hmm. that PD-1 is not the only negative co-stimulator. So could there be other effects as well?
Yes, that is true. And in line with sepsis being marked by pro-inflammatory and anti-inflammatory effects, we also found some anti-inflammatory molecules that respond to miR-93, for example, CD160, which is a co-inhibitory molecule for this kind of signaling. So also miR-93 seems to actually target both pro and anti-inflammatory components in these T cells. So the way I see it, it should be like a delicate balance that needs to be achieved in these T cells to get the proper function and not get too much immunoparalysis in these patients. Mm-hmm.
I don't see any other open questions at this point. I think we've addressed all of the ones that were there. Mm-hmm. What a great attendance. I think at one point I saw almost 70 participants today, and really great job to both Melanie and to Elaine for presenting really provocative talks. And I think the speakers, but also the participants, this RNA Collaborative series has just been outstanding to really bring the community together. And it started off during the pandemic, but I think it's a great way to keep the community going together. Again, thank you all for participating today. Grace, I'll turn this back to you.
All right, thank you. We're going to end now. Thank you, everyone.